Hello students, today's class will be salivary glands from oral histology. Salivary glands, in this class we'll be covering growth and development of salivary glands, structure of salivary glands, ductal system and the connective tissue of salivary glands. Growth and development of salivary glands, they arise from the ectoderm of the oral cavity. One word to note, salivary glands arise from the ectoderm of the oral cavity. Other ectodermal derivatives are sebaceous glands and mammary glands. The first evidence of appearance of a salivary gland is 6th week of intrauterine life. That is, the parotid and the submandibular gland arises at the 6th week of intrauterine life. At the 7th week of intrauterine life, sublingual gland arises. Minor salivary glands arise at 3 months of intrauterine life. There are 3 stages in the development of a salivary gland. First stage is analog formation with development of the gland along with dichromatic branched ducts. Second stage will be differentiation plus formation of lobules and canalization of the duct. This stage will last till the 7th month of intrauterine life. The third stage is maturation and acinar and intercalated duct differentiation. Kindly note down these 3 stages. We'll come again. First stage is analog formation with development of the gland along with dichromatic branches. Second stage, differentiation and formation of lobules and canalization of the ducts. Third stage is maturation plus acinar and intercalated duct differentiation. Structure of the salivary glands we are going to see, and in the structure of salivary glands we'll be covering the following topics in detail: acinar cells, tight junctions, central lumen, serous cells, mucus cells, myoepithelial cells and ductal system of salivary glands. Now pay attention students, because a lot of MCQs have been asked on the structure of salivary glands in the previous year papers. Speaking about acinar unit, what is acinar unit of salivary gland is? Acinar cells, they are the basic functional unit of a salivary gland. They are made up of epithelial secretory cells. Now these epithelial secretory cells are of 3 types: serous, mucus and myoepithelial cells. So acinar cells are the basic functional unit. They can be made up of 3 types of epithelial cells, namely serous, mucus and myoepithelial cells. Acinar unit, this is the image of acinar unit. Acinar, if you can see, acinus. Acinar unit means nothing but where the saliva production starts, or the unit which is responsible for saliva production and ejection is acinar unit. Acinar cells are arranged on the basement membrane in a single layer. If you can see, serous cells or mucus cells and the ductal system. This area is the acinar unit. It can be made up of serous, mucus or myoepithelial cells. This circular area, pointer is pointing here, and this circular area is the acinar unit. The acinar unit is followed by a ductal system, which we will be seeing later. Now, tight junctions. Interacinar spaces and the lumen are separated by 3 types of tight junctions. You can see in the previous image, interacinar spaces. This is 2 acinar cells. Interacinar spaces and the lumen. Lumen is the central area. They are separated by 3 types of tight junctions. They are zona occludens, which is tight, zona adherens, intermediate, and macula densa, which is 1 or more desmosomes. Function of tight junctions is it maintains the polarity and homeostasis. Tight junction proteins are, please take a note, claudin, occludin, and junctional adhesion molecules. You can see in this image, this double line between these 2 acinar cells is the tight junction. And between the lumen and the acinar cell also, you have this double line. They are the tight junctions and they are of 3 types. Their function is to maintain polarity and homeostasis. And tight junction proteins are claudin, occludin, and junctional adhesion molecules. Central lumen. The central lumen is star-shaped. If you can see, leading to the lumen, their mucous acini lumens are larger when compared to serous acini lumen. If you can see in this image, you can appreciate lumen well. This is a lumen. The central area between the acina cells is the lumen. It is usually roughly star-shaped, and they are usually larger in a mucous acini compared to a serous acini. We'll see about mucous and serous acini in detail. Serous acini, they are pyramidal in shape. You can see this image, students. Pyramidal in shape. This is the magnified diagram of a serous cell, serous acina cell. Pyramidal in shape, spherical nucleus at the basement membrane. They are usually darkly stained and they have presence of secretory granules. How to identify a serous cell? They are pyramidal in shape. If you can see in this area, they are pyramidal in shape. They have a round nucleus towards the basement membrane and they have secretory granules. They have watery secretion which is isotonic. Serous salivary glands secrete watery secretions and they are isotonic. They are proteinaceous. Their secretion is proteinaceous in nature and they have zygomen granules in the cytoplasm of the cell. Central round nucleus, like we already saw. Serous acini have a smaller lumen and the carbohydrate to protein ratio in a serous cell is less. One word to note, carbohydrate to protein ratio in a serous cell is less, seen predominantly in the salivary glands. Serous cells are seen predominantly in the salivary parotid gland. Moving on to mucous cells. Now mucous cell, the shape by itself is different. Nucleus is oval and flattened and compressed against the basement membrane. When you see a flat oval nucleus, it is a mucous cell. It has a large lumen. Compared to the serous cells, the central lumen of a mucous cell is larger. It is lightly stained. Serous cell is darkly stained. Mucous cell is lightly stained. It has large amount of Golgi apparatus present. There we had secretory granules. Here also we have secretory granules, but here predominantly Golgi apparatus. Demilunes. You have to learn about something called serous demilunes. Now this is a serous demilune. They are nothing but this yellow colored half crescent shaped area. Crescent shaped area is the serous demilune. They are bonnet or crescent shaped covering over the mucous cells, or covering off the mucous cells by the serous cells. That is serous demilune. I repeat again, bonnet or crescent shaped covering of mucous cells by the serous cells. The intercalated junctions are present and the intercellular junctions are present and the carbohydrate to protein ratio is greater. There will be a question, serous demilunes are found in dash. Option A, serous cells. Option B, mucous cells. Option C, myoepithelial cells. The answer is serous demilunes are found in the mucous cells. I repeat again what serous demilunes are. They are bonnet or crescent-shaped covering of mucous cells by the serous cells. Mucous cells are covered by the serous cells. That is a serous demilune. Ductal system of the salivary gland. Now moving on to ductal system, pay attention students. A lot of MCQs have been asked on the ductal system of salivary glands in the previous years. Ductal system of salivary glands is basically of two types, intralobular ducts and the interlobular ducts. Now these intralobular ducts are again of two types, intercalated duct and the striated duct. Intercalated duct is the first ductal system after the acinar unit. The first duct which comes is the intercalated duct. They are low cuboidal epithelium surrounded by myoepithelial cells. Next one is the striated duct. They are tall columnar cells with large central lumen with a prominent striation at the basal end. It has an important function, makes saliva hypotonic, that is, water resorption and ion secretion takes place here. Second is the interlobular or the excretory duct. It has two layers and it is the larger excretory ducts you can find goblet and cilia cells. It has pseudostratified ciliated columnar epithelium. Intercalated duct has pseudostratified ciliated columnar epithelium, which undergoes differentiation to become stratified squamous epithelium of the oral cavity. You see the image. You can appreciate well in this image also. Intercalated, striated, and the interlobular or the excretory ductal system. Intercalated duct is a low cuboidal ductal system. Striated are tall columnar with striations at the end, and they are responsible for making the saliva hypotonic. Then the excretory duct is large. It has goblet cells and cilia. And these excretory ducts are pseudostratified columnar epithelium. Connective tissue of the salivary glands. It contains fibroblasts, macrophages, mast cells, leukocytes, fats, plasma cells, collagen, reticular fibers, ground substance consisting of proteoglycans and glycoprotein. Now, something to pay attention about connective tissue of salivary glands is that it has a capsule. If you can see in this diagram, connective tissue of the salivary gland has a capsule which extends as septa. These light colored areas are the septa. So capsule extending as septa into the connective tissue is a feature to note in the salivary gland. That's it. We are ending this session of salivary gland. MCQs. The first evidence of appearance of salivary glands is option A, 6 weeks of intrauterine, 8 months of IUL, 3 months of IUL, and 1 month of IUL. IUL is intrauterine life. First evidence of appearance of salivary gland is 6 weeks of intrauterine life. And the salivary gland which we see is parotid and the submandibular gland. Second MCQ. Striated ductal system of the salivary gland is lined by dash epithelium. Options: low cuboidal, tall columnar, pseudostratified ciliated columnar epithelium, and stratified squamous epithelium. Answer is striated ductal system of the salivary gland is lined by tall columnar epithelium. We saw 3 types of ducts: intercalated, striated, and interlobular. Intercalated is lined by low cuboidal cells, striated tall columnar cells, and interlobular or excretory duct is lined by pseudostratified ciliated columnar epithelium. That's it for this class. Thank you.